So how do you understand this simple t-squared law? Well, I want to give you sort of a number of variants on it here, but it's really simple kinetics phenomenology that gives it to you. Okay. So the basic picture is that if you plot free energy as a function of aggregate size, so here would be 1, and here would be some big number, maybe 10 over here. And somewhere in the middle, there's going to be some critical nucleus size. So we measure everything relative to 1, and then there's a barrier here which is of the order of a few kilocalories per mole, maybe 1 to 2 or 3 kilocalories per mole, which has a metastable minimum at this nucleus size right here. And then if you can get over that barrier, then the things just run away because you like to form large beta aggregates. So if you can get over that barrier, then things like to run away. You might eventually reach an equilibrium as long as you can peel monomers off of those aggregates. But that's the basic idea, that you have to get over this barrier and then things can run away. And so the picture is that we have some initial concentration of these nuclei that can lead to the aggregation. And there's two ways we can imagine coming up with some initial concentration of these nuclei. The first way is we can imagine that we simply put them in. That is, we seed a solution with nuclei, and then we'll just say that that's just whatever concentration we put in. So in that picture, we have some monomers in our solution, and we have a few nuclei in our solution that we just put in there to begin with. Maybe we've cultivated those from in vivo from live tissues. The other possibility is that there is an equilibration there's an equilibration then between the monomers initially and the nuclei initially. So that after we put the stuff in solution, reach this initial equilibration of monomers and nuclei, then after a while you start to elongate the nuclei into the aggregates. So there's two ways then to think about this, and we'll examine both of those. Now, there are then two quantities of interest that we want to write down simple kinetic models for. And then I'll do a couple of slight tweaks to these kinetic models so you can see other features. First off, Cp is the concentration in polymer concentration of polymers, not distinguishing what length those polymers are. It's just the total concentration of polymers in solution. And delta is the number. Delta is the basically the total concentration of monomers in polymer form. Okay, and then this is what you measure in these experiments here, where thioflavin T is this dye that only attaches to beta amyloid. sorry, that only fluoresces when it attaches to beta amyloid. In fact, it's really not known exactly why that works. So there's a problem for people to look at. Why is it that thioflavin T will fluoresce when it attaches to amyloid? But it is known empirically that if you attach thioflavin T to amyloid, you can get a fluorescence. And so by measuring the fluorescence intensity of the thioflavin T, that will give you the amount of polymer, the amount of monomer in aggregate form, because it attaches monomer by monomer, shown in the upper panel right there. It's done in the experiments by Ferrone, Chen, and Wetzel. Okay, so this is what's measured, for example, by thioflavin T fluorescence. Okay, so the equations are really simple. You write down that the rate of change, and we're going to assume that Cp at time t equals 0 is 0, and that's equal to delta at time t equals 0. So we're either going to grow these polymers either by seeding with some nuclei or by allowing those nuclei to come from equilibrium, from a weak equilibrium with the monomers. Okay, so dCp dt is equal to K plus times C1 times C, the concentration of the monomers. So this is to say that we have a rate constant for adding monomers to nuclei. So this is the rate constant for adding monomers to nuclei, and the concentration of the nuclei is given by this. Okay, and then the rate of growth of monomers in polymer form, well, that's equal to, we grow monomers in polymer form by adding monomers to existing polymers. So that tells us that in the simplest model, that the growth of monomers in polymer form is just by taking the total concentration of polymers and then adding monomers to those polymers. So we add monomers to nuclei to grow new polymers. We add monomers to polymers to grow more monomers in polymer form. And again, this is really the experimentally interesting quantity right here that you pick out by the thioflavin T dye. Okay, so right away you can see something interesting here. Right away you can see that at short times from the start of the experiment, we can take and replace C1 by the initial concentration of monomers and Cm star by the initial concentration of nuclei. However, we put those nuclei in there, whether we let them come from a weak equilibration or we let the nuclei come from seeding of the solution. And right away what we can see is that given the initial conditions, that Cp of T is just equal to K plus C1 naught Cm naught star times T. That is, the number of polymers just grows linear in time. And that delta of T just grows quadratically in time. So this is K plus squared, C1 naught squared, Cm naught star times T squared. And of course, that's the T squared that's showing up in this graph right up here. Now, then the question is, what's the condition in the experiments right here? What's the condition that gives rise to the aggregation? Well, in these experiments, there's no nucleation. Sorry, in these experiments, there's no seeding. We're not putting in the nuclei in order to start the experiment running. We're letting the nuclei form as the experiment runs. So we're in this condition right here. We're in a condition of equilibrium, weak equilibrium right here. And so what that tells us is that if we're in this condition of weak equilibrium, so we're in the no seeding condition, which is what we have in the experiment, then there's something really quite interesting that's experimentally useful that comes out of this, this experiment. Namely, we can see that the rate of growth is equal to this equilibrium constant times K plus squared times C1 naught to the m plus 2, where we've substituted in times T squared, where we've substituted in that Cm naught star is equal to Km times C1 naught to the m power. So what that says is that if we vary the concentration, we can actually see what the critical nucleus size is. So by doing plots at varying concentration, we can then read off what the critical nucleus size is. And interestingly, in this study of Chen et al., when they vary the concentration for each one of those sizes there, they find that the critical nucleus is 1. So for the poly-Q diseases, at least at these links, the critical nucleus seems to be 1. You seem to be getting some kind of a beta structure already for just a monomer, which is pretty interesting. Some kind of a nuclear beta structure already for just a monomer. Now, this doesn't actually prove that it's beta. It just proves that it strongly suggests that you just need one monomer to get it going. That monomer could start out perhaps being a little bit disordered and then harden once it gets into aggregate form. That much we don't know. But it does strongly suggest that you need one monomer. Now, just to add a little bit of intrigue to this, just in the last year, Ron Wenzel has done follow-up experiments on this, and he's shown that this critical nucleus size is very dependent upon the length of your aggregates. And in particular, if you have a Q length which is less than about 23, then that critical nucleus suddenly pops up to something like 4. So it's very dependent upon how long your Q is. So it's not just that the Q length determines a slowdown of the disease, it's also that the process changes as you get to smaller Q lengths. And that means it's even harder to get the disease if you have a much smaller repeat length inherently. Okay. So let me just add, so is this, any questions about this? So this is really nice. I mean, I've got to put caveats on it. Not everything is elongation. It works in the test tube. It may not work in the body. Not everything is elongation. I'll show you one example of where it works in the body here in just a second. But, and it is linear elongation, okay? And so if you have these oligomers or if you have nonlinear oligomerization, that is non-one-dimensional oligomerization, then this is not going to be a good description of the process. But there is this belief that the aggregation processes we observe in the test tube probably have at least something to do with the aggregation processes in the body. So hopefully one can use this. And I think this is a very useful and simple phenomenology. Maybe let me show you this example of where the T-squared really seems to occur in the bodies of mice. So one of the mammalian prion protein in its normal form is a membrane-bound protein. It lives on the surface of the neurons, and it's recycled continuously. It goes to the surface and comes back. And in order for that to happen, the mammalian form of the prion protein has a so-called GPI anchor. It has a glycophospholipid that attaches to the C-terminal of the protein. And then the N-terminal, which is basically disordered, and the N-terminal, which is the part that's implicated in the aggregation, the N-terminal will float away from that. So the C-terminal has a GPI anchor which attaches into the membrane. So several years ago, a group of collaboration between the Rocky Mountain Institute, looking for, there you are, great, between the Rocky Mountain Institute and Scripps, they got these transgenic mice in which this GPI anchor was deleted. Okay. Then what they did was they inoculated those mice intracerebrally. They took infectious prion material and inoculated those mice intracerebrally. They found that those mice would aggregate infectious prion material, but the mice never came down with the disease. So this GPI anchor is somehow critical to the toxicity of the disease. And so a simple picture is that if you don't have the GPI anchor, then you just sort of send those prion proteins out in the intracellular medium like a spacewalker who's lost their tether off of a spaceship. And then if you put in seeds of infectious material, then you can grow more infectious material, but you can't bring it back into the cell to kill it. So the GPI anchor is somehow critical to bringing things back into the cell to kill it. We also, one aspect of prion diseases, which is different than every other amyloid disease, is that the prion disease also has an infectious exponential growth inside of organisms. It's the only, well, actually there's some belief that all of these diseases are infectious in the sense that if you get a seed of Alzheimer's disease, unfortunately, into your brain, or a seed of familiar Lou Gehrig into your brain, that you can probably come down with the disease. This is actually a real worry, and a lot of groups are focusing on this issue right now, including Prusner has shown recently that there's a seeding spread of A beta from cell to cell inside of brains. Prusner, who won the Nobel Prize for prion research. But there's another aspect to infectivity other than just seeding. That's also exponential replication. And the prions are the only disease of this amyloid form where there's exponential replication. In fact, if you take a look at the data, which is shown up here for these mice, it turns out that this data for these transgenic mice showing the growth of infectious prion material inside of these mice. Forget about this, this is a little more complicated to explain, but this actually just obeys the same t-squared law that we saw from this analysis right here. Oh, I'm sorry, actually this is interesting. So the wild type mice, if you infect them with the same amount of aggregate, they show a very slow growth and then an exponential takeoff, and then a saturation as you start to kill the cells and deplete the supply of ordinary prion protein. So this would be the growth of infectivity in wild type mice. This is the growth of infectivity in these transgenic mice. It just shoots right past this wild type limit. And if you look at that, it's just growing like t-squared. In fact, what finally kills these mice is that they get too much amyloid in their bodies, and it actually damages their heart. It gets into the bloodstream and it damages their heart. That's what winds up killing these mice. So if you take that data, you can put it on a nice regression plot to a t-squared fit. And the idea is that we're putting in these nuclear seeds from the infectious prion material that we inoculate in the brain, and we're just getting linear elongation of those seeds in the intracellular medium. And so we could show that you can get a nice t-squared fit. You can make some estimates then of what the prion concentration is inside the body. And that the prion concentration of the transgenic mice intracellular is about 20 times the prion concentration of the wild type mice. So you can actually, you build up prion concentration because you no longer can clear the prion off the surface. You can no longer take it off the surface by the usual mechanism.
Sorry, I don't know if I understood, but without the molecule in the membrane, polymers cannot elongate. Monomers cannot form polymers.
Well, with the membrane, you can still potentially polymerize on the membrane. But what we're showing here is that the membrane also somehow mediates the exponential growth. So because this t-squared behavior that goes over such a long period is exactly what we see back here. Let me put the wild type, oops. Okay, so that's just these data points right here. But if you put the wild type mice on, the wild type mice would have a lag time and then this rapid exponential growth of the prion material and then saturate because you're killing the cells and robbing the supply of prion monomers. So in fact, you're getting linear elongation, you're getting polymerization, but you're not somehow recycling it back in, and you're not exponentially replicating it. So the membrane does two things. The membrane mediates the toxicity of the prion protein, and the membrane also mediates the exponential growth somehow. We don't know what mechanism is there for the exponential growth yet, but whatever it is, it has to involve the membrane.
Monomers can join each other and form disaggregate structures.
Right, so the monomers can attach on the membrane also. You can grow aggregates on the membrane as well. That's actually what you seem to need for toxicity and for exponential growth. What this is showing is that these transgenic mice, you're just aggregating outside the cells. So you're still generating infectious material because those seeds that you put in grow more of the same stuff. So if you take the brain matter from those transgenic mice, you can infect wild type mice, still get prion disease. But if you but you don't get the prion disease in these transgenic mice. They'll just die from too much protein aggregate. Yeah, I think yes. I should probably sort of wrap this part up Okay, so I'll continue with some discussion of kinetics in the second lecture, which is later today. And maybe are there any other questions you might have? We have a few minutes for questions. Yeah.
So you consider monomer building on monomer, and then the monomer adding to a polymer.
Right.
But what about polymer to polymer?
That's a possibility
Theoretically.
also, but that's a little bit, you know, then you have to try to take these sort of Titanics and
Right.
weld them together. But just a comment maybe, so that's possible, but that would be you could add that in, but that's probably a small contribution. The main growth of the polymers is by rapid addition of monomers.
Linearly growing?
Yeah.
Like that?
Yeah. Now I should say that the same critical concentration, and I'll have to get to this in the second lecture, the same critical concentration idea that we got out from the simple free energy analysis, that's also in principle in the kinetic analysis, but in order for that to happen, we have to allow an equilibration between the monomers and the polymers. So if we allow the monomers to pop back off, then we'll also see a critical concentration emerge that basically has the same form. Okay, so that's one comment. Another comment is the exponential growth. There's basically three ways of getting the exponential growth. One is you can break up the aggregates. You can make them fission. So for example, if you grow aggregates on the surface, and then because of surface undulations or maybe formation of vesicles that enclose some of the prion protein, you might actually break the aggregates. If you break the aggregates, it's sort of like nuclear fission, and then you can have exponential growth off of those broken fragments. Another way that's not quite what you're talking about, but is if on a given aggregate you grow branches, and those branches can then pop off, then the number of new polymers will be proportional to the number of old polymers, and you'll get exponential growth. And finally, the third way, which was believed to be what was important for the prion protein, is really genuine autocatalysis, where you have some entity come in, replicate itself, and then pop apart, and then that's autocatalytic. At the monomer level, that won't work for prions. It can be shown that it's just not at all plausible. But you could have oligomeric autocatalysis, and that could actually be plausible for the prions. Whatever it is for the prions, the exponential growth has to involve the membrane somehow. That's what these experiments with the transgenic anchor, the CGI anchor removed. That's what these experiments show. Other questions?
You're talking about you need the GBA protein to take the polymer from inside the cell, to take the outside the cell to inside the cell.
Yeah, you need it. The problem is you can certainly take the prion outside the cell when it's produced.
Yeah.
You do that through exocytosis.
Mm-hmm.
But then the problem is if it's not bound to the membrane, it just floats away.
Okay.
Right?
So the GPI is not involved in the aggregation itself.
Well, it's not necessary for the aggregation in the sense that if you put the seeds in, you'll get aggregation. However, it's somehow necessary for the toxicity. Right. Yeah, Kirsten.
Yeah, maybe we'll talk in a second lecture. I mean, how about if we include an isomerization reaction? So we have the protein, so it needs to become this potential to, go into a misfolded conformation. So how do you work with the equations? I mean, and that will affect a lot of the lag phase.
Yeah. Yeah, so that would say that you have to get the monomer into a different conformation in order for it to. I haven't put that into kinetic equations, but I will give you an example of that in the second talk that involves the mammalian prion protein, at least a speculation about it for the mammalian prion protein. So, but you could certainly put in an extra. You could have a concentration of the isomers, and it's the isomers which then drive the. Right. Yeah. Yeah.
Is there some work talking about interactions between GPI and an aggregate from prion or something? You know, how is the interaction between these two, the aggregate and GPI itself?
Well, at least if you take, and I might stand corrected from the Rocky Mountain lab, but at least if you take brain isolate from wild type mice that have been infected, then in principle all the infectious prions themselves should have the GPI anchors on them still. So you'll still have the GPI anchors on the infectious prions. So really, I guess this unusual one is just showing the importance of the membrane in this case to mediate the disease.
Yes, it can be another receptor involved in this mechanism too. You know.
Oh yeah, well actually that's been speculated in fact.
I mean, I don't know if I'm right.
No, there has been some work on
GPI
that.
is a protein
Right.
in the inner membrane. But you have interaction in outer membrane.
Well,
right?
but it's even when it's on the outer membrane, it
Oh.
has a GPI anchor. So even when the normal prion is on the outer membrane, so it goes out there by exocytosis. In those vesicles in exocytosis, the GPI anchor embeds it into the membrane, and then those just open up, and then it's sitting on the membrane. And then in endocytosis, you just
Oh yeah.
close it back up, right? But so the GPI anchor is there regardless for the normal prion protein. However, there are other receptors that have been speculated, receptors potentially for the infectious prion protein. How good the state-of-the-art understanding is of that, actually, I don't know, but maybe again from Rocky Mountain Lab we know.
Discussing that.
You'll discuss that, good, okay. Yeah.
Thank you.
Yes?
Aggregation process has a polarity. I ask this because neural cells
Yes.
are polarized.
Uh-huh.
You explained that near the membrane are that important that aggregation is linked to the membrane.
Uh-huh.
I feel really is the toxicity.
So I'm sorry,
related to polarity in the process of aggregation.
So you're saying, let me see if I understand, the polarization of the membrane, is that you're saying, is it known whether the polarization of the membrane is somehow important for the toxicity? Is that the question? Or did I misunderstand?
That if aggregation process has a polarity itself.
Okay. Oh, you mean if you get polar aggregates? These are non-polar. It's not like actin or microtubules. These are non-polar aggregates. I mean, there are some charges on here, but there's no particular polarity to the aggregates here. That's a good question, yeah. So it's unlike the cytoskeletal elements, for example, right?
And is there any order in which these monomers interact, or they can interact in many ways?
I'm not sure I understand your question.
I mean, is there
Are there other modes of
how
self-assembly?
the monomers interact to form these polymers?
Oh, right.
Is
Well,
there any specific way, or there are many ways?
well, in this particular case, at least the envisioning is that once you form a nucleus, you've got sort of exposed beta strand surfaces, and then you just bring up new monomers to those beta strand surfaces. If it's the beta helix, it's really easy. You just lay it down and then you just start to coil it on. If it's the beta sheet, it's not so hard. You just sort of do that. So in this case, the picture is that you just begin to aggregate the beta. But let me say there are some people who are exploring the idea for poly-Q diseases that you first form a sort of disordered aggregate, and that you then sort of harden the beta structure within that. If that happens on a relatively if the hardening happens on a relatively fast time scale, you're not going to be able to tell the difference in these kinetics experiments. So that's a possibility that's being explored for the poly-Q.
Just because I go with big proteins, and they are many times they are disordered, and they have a couple folding related to the recognition.
Right.
And I see that maybe this could be something similar, because you have disordered monomers,
Right.
and without the GPA, was it?
GPI, yeah.
They cannot form these polymers quickly or in a exponential
Oh, right, I see.
way.
Right,
Is
right.
it something similar?
Let's see.
Does these monomers have how you say, maybe could form some disorder polymer or
Right.
structure, and then it falls up if they
Yep.
have the GPA?
Yeah. Whether that might happen, in fact, that might happen with the prion. In fact, there's some evidence of sort of non-ordered prion aggregates near the surface. Whether that's applied to the function. Now, I'm just thinking for the P53, where the functional form of the P53 is a tetramer. That might actually be something along the lines of what you're describing. Are you going to talk about that? So we might hear more about that from Gerson's lecture. Yeah. Let's see. Yeah.
You say, well, on your work that beta strand prone aggregation, but it's also possible
like interconversion of
Yeah,
alpha
the
to
alpha structure was the sequence to prove the aggregation.
Yeah.
So it's possible there haven't.
Again, yeah, I guess it's possible. There are some designed peptides which actually, with pH or with something else, you can switch between an alpha form and a beta form.
Yeah, but.
Right.
Yeah.
Then if you get to the beta form, then obviously they can
But you have
aggregate.
to have the beta structure just to have the amyloid aggregation. You can't have the alpha structure.
Well, in that case, for example, if you brought two of those peptides together,
Yeah.
right, that there might be some reasons for hydrophobicity or something that they'd like to come together. You can design them that way, and then you could flip the switch, and then you form a beta aggregate
Okay.
after.
Okay.
Yeah. So it's certainly, I wouldn't say it's outside the realm of possibility. Nothing comes to mind immediately for that, but it's not outside the realm of possibility.
Oh, okay, okay.
Particularly with these design peptides, you could do that.
Oh.
Yeah.
Do you think that is the beta super topology who start all this process? Or because you were talking to that the oligomers start the disease,
Right.
But you have monomers and polymers outside the cell. But you think that's beta super topology? It's that who starts the process of the pagination of the.
Well, that's a good question.
Polymer.
So again, certainly in the case here, we know that those prion, infectious prion agents that we seed with, they have cross-beta structure as inferred from dichroism and from the fibrils that they form. Okay, so those can also be formed in two-dimensional crystal structures, and maybe I'll say a little bit more about that in the second lecture. So we know that the cross-beta structure is present there. But getting back to this question, I think we'll probably hear a lot more from Christian about this, it may not even be the beta structure that causes damaging oligomers, okay? It may be that you can have the oligomers come together and that you sort of harden those into a beta structure once the oligomers come together. And therefore the oligomers might have something different going on. So I think that question is wide open. That's one of the questions that we'd love to have answers to. Yeah, yeah. So I probably should break now. I actually have to go use Adobe Connect to give a lecture to my class at Davis, so I'll be absent for a little while here. But thank you very much for the great questions and for your attention.
Okay, thank you.